Okay, we are recording, so I'm going to get started really fast. This is chapter 5 in your textbook, in your Pearson textbook. I'm going to do just a quick blast review of the skin, the layers of the skin. So let me share screens, collect my whiteboard. Actually, I'm not even going to do that. I'm just going to remind you there are 5 layers to the epidermis, and from deep going to most superficial, it goes stratum basal, stratum spinosum, stratum granulosum, stratum lucidum in thick layers of the skin, which are the palms of the hands and the soles of the feet. That's the only place we find stratum lucidum. And then finally, the 5th most superficial layer is the stratum corneum. I didn't mention much about the hypodermis in our first lecture, so I do want to say a few things about that. The term hypodermis literally refers to what's below the skin. So the hypodermis technically is not part of the skin. It's not. It's deeper than the dermis. So if you are asked what is the deepest layer of the skin, the hypodermis is not it. The hypodermis is not part of the skin. Actually, the reticular dermis is the deepest layer of the skin. This is under reticular dermis. And the hypodermis is made up mostly of of fat cells. So I'm just going to write fat, adipose tissue. I also wanted to define with you the differences between, this is not really, in my opinion, made all that clear in the textbook. There are 2 types of tactile receptors or cells in the skin. And I wanted to be extra clear about the differences between the 2. Merkel cells, that's M-E-R-K-E-L. These are technically found very close to the junction of the epidermis and the dermis. And so they sort of float around near the deepest layers of the epidermis. These are actually cells that are attached to nerve endings. And these tend to respond to light touch. Okay. There's a different type of tactile element. It's technically not a cell, so I'm going to put versus Merkel cells versus Meissner's corpuscles. M-E-I-S-S-N-E-R-S, just named after the guy who first discovered them. Corpuscles. So technically, these are not cells, individual cells. Meissner's corpuscles are located deeper than Merkel cells. So these are found within the papillary dermis, not at that junction. In the papillary dermis, mostly. And these are they're just free nerve endings. And when we study the nervous system and study sensory receptors, I'll probably draw this again in more detail. Free nerve endings are just the end of a nerve fiber that ends in a coil like this. It's not really an individual cell itself. These tend to be for deep touch, not light touch, but for deep touch or pressure. So I'm going to write for deep touch / they're also, types of them are pain receptors as well. I want to review that. This term, flexure lines, briefly talked about this topic, I think, before we left the first lecture. These are what the definition of flexure lines, they're dermal folds. So down in the dermis, there are folds. And it has to do with the collagen fiber arrangement. Collagen fibers give the dermis, its toughness, its strength, and its ability to resist abrasion. But they're bundled together, these fibers. And sort of at the areas where there's a break in the bundles, we call these flexure lines, and these are typically at or near our joints. And it helps facilitate movement, usually at that joint. So we're not fighting against these collagen bundles when we try to move a freely movable joint. Okay, new topic. So I'm going to go on, talk about skin color. We did hit melanocytes when we talked about the stratum basale, but there are a few other factors that determine our skin color. Basically, there's three main factors. Melanin is the big one. There's also a pigment called carotene, probably heard of that, and hemoglobin, especially in fair-skinned people. So let's talk about these individually. We know a little bit about melanin. This is a pigment that's secreted by melanocytes. We already went over that. What it is, it's a polymer made from the amino acid tyrosine. So one of the amino acids that we find in the human body is called tyrosine, and that's the basis for the pigment melanin. There's two different types of melanin, by the way. So I'm just going to briefly mention that. One tends to be kind of yellow-red in color, and the other tends to be brownish-black. There's an enzyme in our cells that causes the conversion of this amino acid into melanin. So the enzyme that regulates the formation of melanin from this amino acid is called tyrosinase, just gets its name from tyrosine. This enzyme regulates the synthesis of melanin. Remember I mentioned in our last lecture, we all have the same relative number of melanocytes in our skin. Doesn't matter what our skin color is, we all have about the same amount of melanocytes. Differences in skin color, as I said, is determined by how active our melanocytes are and how actively they produce these different two types of melanin. That's it. So UV light is what hits the deeper layers of the epidermis, and it energizes really the pathways, this keratinocytes that are newly formed by the basal stem cells. They release chemicals that help activate this enzyme, and then this enzyme converts this amino acid into this pigment, this polymer pigment, and that's how it's produced. And then it oozes around in those deeper layers of the epidermis. It incorporates itself into those newly formed cells by the basal stem cells, and it helps protect them from sun damage. Second, so we've talked about melanin. Let's mention a few things about carotene. Carotene is another pigment. It's yellow-orange in color. We find this pigment in plant products that we ingest. So I'm just gonna say found in food, then in parentheses, I'll just put plants. It's where carrots get their name. So carrots are obviously high in this pigment, this yellow-orange pigment. Other foods are too as well. Egg yolk has some of this pigment in it. This accumulates, unlike melanin. Melanin, remember, is synthesized and found deep in the epidermis, down in the stratum basal, a little bit in the stratum, and it stays down, our melanin stays down in those deeper layers of the epidermis. Carotene tends to, when we ingest a lot of it in our diet, it tends to collect in the most superficial layers in the stratum corneum. So we find it closer to the surface of the skin. It also can accumulate a little bit. I'm going to put some in the hypodermis. It also tends to accumulate in the adipose tissue, fat cells of the hypodermis. Okay. And finally, hemoglobin. I'll do that on my next page. These three things contribute to our skin color, mostly melanin, but then carotene, probably in the order that I'm covering them right here. Hemoglobin is really, it's a protein. It's a globular protein that binds oxygen in our red blood cells. That's its main function, is that it's an oxygen binding protein that we find in our red blood cells. It carries oxygen to our tissues in the body. But when oxygen binds to hemoglobin, it turns our blood a bright red color. It turns those red blood cells bright red from a dull red. So we find hemoglobin everywhere that we find blood. It's carrying oxygen, but particularly in relationship to skin color, dermal capillaries, that just means small blood vessels in the dermis, carry O2-rich blood. So the blood vessels that are closest to the surface of our skin, is what this statement is saying, can influence skin color, particularly in fair-skinned people, to a reddish color. So it's more obvious in really fair-skinned people. My parents were both from Scotland, so I'm fair-skinned and I'm very subject to sunburn. And when fair-skinned people in particular, like you exercise, it increases blood flow to the surface all over the place, but particularly in surface areas of the skin, that's to release heat. And that blood flow, that increased blood flow to that area also causes a reddish hue to be seen. We find this it's most influential, not just in fair-skinned people, but on the palms and soles, because that's where the most of the surface circulation in the dermis is found. All right, let's be done. We'll call that good with skin pigmentation or skin color, the three things that influence skin color. And I want to say a few things about different hair types. Okay, so these just it's just names of different hair types, and there's two main classes. Vellus, V-E-L-L-U-S. Vellus hair tends to be the super fine, what you and I call baby hair, because when we're first born, when all humans are first born, they're covered with this little tiny super fine hair all over their bodies. That's super fine. I'm going to put baby hair in quotes. That's very different type of hair than terminal hair. Terminal hair tends to be coarse, has more color in it as well. So I'm just going to write coarse, much darker because it has more pigment, more melanin in it. And where we tend to find terminal hair is on top of the head, where you and I grow our hair and get it cut. In males, in the beard, facial hair in males. I'll put that in parentheses. Pubic hair in the groin region and the axillary region of the armpit. We call this type of hair terminal hair. Now I'm going to switch gears again. I'm trying to hit all of the topics, as many as I can, in chapter 5 before we cut this off. And this is a little bit longer topic on sweat glands, so I'm not going to waste my whiteboard space. I'm going to begin a new topic here. These are also called sudiferous glands. Sort of the scientific language of sweat glands is sudiferous glands. We find these, I'll give you some fun facts about them, then I'm going to go into the specific types of sweat glands that we have in the human body. We find sweat glands pretty much on the entire body. Except on the nipples of the chest and external genitalia. In both males and females. Roughly, that's an average sign, or about, we have roughly about 3 million covering our body. Now I need to go to a new whiteboard. There are 2 major classes or categories of sweat glands. And there are some subcategories within those 2 types. First, number 1, it's pronounced eccrine, E-C-C-R-I-N-E. Eccrine glands or eccrine sweat glands. These are merocrine in secretion. We define that, what merocrine is. I'll remind you right now. Merocrine is a gland that releases its secretion by exocytosis, and the secreting cell does not get damaged in the secretory process. So that secretory cell can secrete over and over and over again. So the type of secretion is merocrine in nature. These are the most numerous on the human body, eccrine glands. So we find them all over the place. How they are arranged, they are just a simple coiled, getting ahead of myself there, a simple coiled duct. Try to draw you a picture of that opens onto the surface of the skin. So really quickly, if this is the surface of the skin, a typical eccrine gland would look like this. And then it's a tube that just coils at the bottom, and then singular tube up to the surface of the skin. The secretion, as I said, is released by exocytosis in a merocrine nature. So the secretory cells that are really found down here at the base of the gland are not damaged in the secretory process. And now I'm going to tell you what's actually in. So I'm just writing secretion, what's in the sweat itself. It's about 99% water. So it's a water-based product. There are some salts found in this, so I'll put that in parentheses. And it can be a minor way for us to excrete some salts from the fluids in the body. We also find some vitamin C in these secretions, particularly if we are loaded on vitamin C internally. If we've ingested an abundance of vitamin C and our fluids tend to be overloaded with it and our cells aren't using it, we can get rid of excess vitamin C in these secretions. There are also antibodies, which of course are antibacterial in nature. Dermacidin, this is an antibacterial peptide. It's not necessarily an antibody. Antibodies are protein-based, so this is just a small little peptide. It behaves like an antibody. Peptide just means a small chain of amino acids. That's all that word means. Occasionally, we might have some waste product, things like ammonia, urea. Those are from breakdowns of nitrogenous bases, nitrogenous-based products. And the last thing I'm going to say is this sweat tends to be slightly acidic. That also helps fight bacterial growth on the surface of our skin as well. These glands, these sweat glands, these eccrine sweat glands are regulated by the sympathetic nervous system. When we talk about the nervous system at the end of this semester, I'll revisit this. It's the part of the autonomic nervous system. That means we don't have conscious control over it. It's a part of the autonomic nervous system that is when we're stressed out, basically, when our bodies are stressed either physically or we are emotionally stressed. It's a fight or flight response, this sympathetic nervous system response. So you know that you start to sweat when you're physically stressed, like when you're exercising, and or when you're emotionally stressed. If you're nervous about a presentation or something, sometimes you begin to sweat. So that's the first major type, eccrine glands. Next page, I'm going to do the second major type. So I'm going to write in number 2, apocrine. Eccrine sweat glands versus apocrine sweat glands. Talk about some similarities and some differences. We have much fewer of these on the human body. I think it's roughly like 2,000. I'm just going to put on the body. These are found predominantly in the axillary region. It's from chapter 1. You should know that that's the armpit region, and axillary and urogenital area. Think the groin area. These are associated with pubic hair. So where we find pubic hair, we find these apocrine sweat glands. Excuse me. These are also merocrine in nature and secretion. So the secretory cells do not get ruptured or destroyed during the secretory process. The glands release their secretions by exocytosis. So the manner of secretion is the same. That's similar to eccrine sweat glands. The ducts of these empty directly into a hair follicle. So that's a main difference. A pubic hair follicle in particular. So unlike the first picture I drew, I'm not going to draw a whole hair follicle over again. By the way, these are different than sebaceous glands. I'm going to address those last. These are just sweat glands that are associated with our pubic hair, and The ducts themselves do not empty directly onto the surface of the skin. They empty into a hair follicle. So that's a major difference. There is some watery fluid. So I'm going to write some water-based. But we have the addition of lipid-based substances and proteins. That's a difference, these lipids and proteins that are found in the secretion. When these lipids and proteins in particular hit the surface of the body, they come up out of a hair follicle, they hit the surface of the body, and then bacteria that are on the surface of our skin actually start to decompose and break down the lipid and protein products. So these, I'm just going to say when decomposed by bacteria, that's what produces body odor. So the secretion itself is not full of odor. It's only after the bacteria on the surface of our skin starts to break down the lipids and proteins in this secretion that produces our body odor. These glands, apocrine glands, become active during puberty, which makes sense because that's when we grow our pubic hair. Oops, misspelling. And that's caused by an androgen release. Androgens are steroid-based hormones. The most common one is testosterone. I'm going to put from, just think androgen is a hormone. These are also activated by the sympathetic nervous system. Trying to squeeze this into the one page. Remember, that's a stress response, fight or flight response. And some people think these are possible, that an analogy or homologous structure to an active sex scent gland in other animals. That's to be debated. So next page, I'm going to talk about a sort of a specialized apocrine gland. They're called ceruminous glands. These are considered to be sort of modified apocrine glands. Okay, they are found in the canal of the external ear. The secretion of these modified apocrine glands, they mix with sebum. Let me write this down. Secretion mixes with sebum, which is that oily base substance secreted by sebaceous glands, to form what you and I call earwax. That has a scientific name. It's called cerumen. That's where they get their name, ceruminous glands. And finally, I'm just gonna, before I get to sebaceous glands, I am gonna just mention mammary glands. I'm not gonna talk a lot about them because in your textbook, mammary glands are covered in great detail when we do the reproductive system very late in A&P2. So these are just specialized sweat glands that secrete milk in mammals. They are considered specialized sweat glands. And you'll learn all about that when you study the reproductive systems in A&P2. This is all I want you to know, that they are considered specialized sweat glands that secrete milk. Now, last but not least, I'm going to go over sebaceous glands. These are technically not sweat glands. So we have eccrine glands and apocrine glands. Ceruminous glands are modified apocrine glands. Mammary glands are specialized sweat glands. So I'm going to just draw a line here, because sebaceous glands technically are not considered sweat glands, but they're associated with each hair follicle. So I'm going to say a few more things about these. I drew them in my previous lecture, a sebaceous gland associated with hair follicles, but these are oil glands that empty into hair follicles or directly onto the skin surface. So they are associated with our hair follicles, but we can find these glands independently arranged, just emptying onto the skin. They are found pretty much everywhere. So I'm just going to put all over the body, except where we lack hair, which is the palms and the soles of the feet. But they tend to be in much higher concentration. So I'm going to high concentration in certain areas of the body. On the face, chest, neck. New page. These sebaceous glands, they secrete an oily based substance called sebum. I'm just going to write oil lipid based secretion. The cells are holocrine in secretion nature. So I'm writing a holocrine secretion, just to remind you what that is. Those cells, the secretory cells, actually rupture during the secretory process. So they're destroyed. Those secretory cells, that's what makes them holocrine glands. And they have to be rejuvenated. New ones have to be created, synthesized, in order to keep the secretion going. Those erector pili muscles that I mentioned in my first skin lecture, their main function is to make our hair stand up on end. Pili just means hair. So erector pili means a hair standing up. Those erector pili smooth muscles, they also assist the sebaceous glands in their secretion. Okay, that's it for glands. I'm going to hit now the 5 major functions of the skin. I'm wrapping up this chapter. So major functions of the skin. I'm gonna just list them and talk about each one. I don't have time to write out a lecture about each one, but I'll talk slowly. And if you need to, just rewatch this recording. First main function of the skin and our integumentary system in general is protection. So I'm just going to list 3 types of protection and say a few things about them. The skin does provide a chemical barrier. For example, the secretions that come up from our skin, we mentioned this in our eccrine sweat glands, are acidic. Melanin is also, I'm going to draw a little number 1 next to this. Melanin is technically considered a chemical that's protective against UV light. The chemicals help control a multiplication of bacteria on the surface of our skin. And defensins are a type of chemical that are also released from skin. So a big, huge protection factor are the chemicals and secretions released by our skin itself. It also mainly provides our first line of defense, which is a physical barrier. Without our skin cells, the epidermis, and partly the dermis, things would just enter the inside of our body. So a physical barrier, and that's formed really by the tight junctions of those keratinocytes in our epidermis. That provides, in the stratum corneum, that provides almost an armor-like physical barrier. Don't forget the waterproofing glycolipids that the stratum granulosum secretes, which helps keep fluids out and helps keep our own body fluids in. So it's a separation physical barrier. And finally, a biological barrier. These headers are in your textbook. I'm just trying to help explain them to you. Biological barrier. We did mention dendritic cells, so I'll put those in parentheses. Those are immune cells that we find in the stratum spinosum. That's that second layer going upward of the epidermis. Those are immune cells, so they provide a biological barrier. They help haul off invading foreign microorganisms for our macrophages to devour. Macrophages themselves are found in the dermis. Those are just gobbling up immune cells as well. And finally, DNA converts UV radiation into heat, which provides a biological barrier as well. So that covers protection. Second major function of the skin is temperature regulation. It helps conserve heat through vasoconstriction. So I'm going to write heat conserving. How does our skin help conserve heat? Well, if we were to walk outside today and it's 30 degrees in a pair of shorts and a t-shirt, not only would our erector pili muscles in our skin help conserve heat by erecting the hairs and causing shivering as well, that's a neural response, but also the blood vessels near the surface of our skin when we are cold constrict. So there's less blood flow going to the surface of our skin to help conserve heat. On the opposite end, our skin can help cooling, predominantly with sweating. The whole act of sweating or perspiration puts fluid, mostly water, on the surface of our skin. And when that fluid evaporates, it cools the surface. So it acts as a cooling mechanism. And we can also get rid of heat via the skin. When we are exercising and we get, I'm putting air quotes up, when we get hot from exercising, the blood flow actually increases to the surface of our skin. And that helps release heat as well from the body. So we've got heating and cooling. Third main function is sensation. That just means we have those coiled nerve endings as well as Merkel cells. So we've got Merkel cells and Meissner's corpuscles that help with light touch, deep touch, pain receptors found in our skin. Let me go to a new page. I'm running out of space here. I'll do 4 and 5. Your book might even do 6. So number 4, metabolic functions. I'm trying to go in the order of your textbook. I jotted these down. Hopefully I did it in the right order. If not, it's all the same. Doesn't matter what order we go in. Metabolic functions from UV light. Remember when UV light generates the formation of vitamin D3. So I'm just going to remind you the formation of vitamin D3 is considered a metabolic function. And then that active form is actually produced in the kidneys, which helps with our calcium absorption in our diet, that long pathway. The skin can also convert, I don't think I mentioned this before, can convert cortisone to hydrocortisone by adding a water molecule to it. And this helps with inflammation. It's anti-inflammatory, actually helps. I'm going to write anti-inflammatory. Number 5, the skin can act as not a major, but a significant blood reservoir. We can hold a lot of our blood, our circulation, the volume of our blood in the skin. Lots of relative term. It's something like 5%. It doesn't sound like a lot, but that's significant. 5% of our blood volume is typically found circulating in the skin itself. That means if we start to bleed, we can use some of that blood reservoir from our skin to stay alive. And last but not least, excretion. In some minor ways, particularly when we sweat, we can secrete small amounts of nitrogenous wastes like urea, ammonia. I mentioned this earlier. Not huge quantities. Most of our excretion is done by the kidney and urine, but some sodium and water as well. All right, guys, that's what I have on my list for today. We can end a little early, which is good. Let me cut off my recording.